Good afternoon, students. Today we are going to start the next topic of this chapter, that is semi-conservative replication of DNA. So let's talk first about replication, right? Okay, so we have studied in the 11th standard about replication and how does replication generally takes place in a DNA, right? Okay, we know DNA is a genetic material and replication of a DNA generally leads to the formation of a daughter DNA, right? Okay, so if you consider any kind of a cell, either it is a prokaryotic cell or either it is a eukaryotic cell, yes, they undergo replication. So here, what exactly is replication of a DNA molecule, right? Okay, replication is a type of a process where replication is a type of a process where DNA molecule generally produces daughter DNA. I can also say that a parent DNA molecule, when undergoes replication, it produces exact replica or exact copy of itself. That process is generally called as replication of DNA, right? Okay, so when I talk about this replication generally takes place in the DNA, and replication of a DNA generally takes place in both prokaryotic cell also, and it generally takes place in the eukaryotic cell also. So let's talk about replication of DNA in a eukaryotic cell. Replication of a DNA in a eukaryotic cell takes place in a cell cycle, right? We very well know what is cell cycle. So it is the cyclic changes taking place in the life of a dividing cell, right? Okay, so replication of a DNA generally takes place in S phase, that is synthetic phase of the cell cycle, where it takes place in the interphase. Interphase is divided into G1 phase, S phase and G2 phase. So replication of a DNA generally takes place in the synthetic phase of the interphase. Okay, now this replication of the DNA for the very first time was generally studied by Watson and Crick, right? You know very well Watson and Crick, that is James Watson and Francis Crick in 1953 were the first scientists for the discovery of the DNA molecule, and same replication of the DNA, that is semi-conservative replication of the DNA, was also given by the scientists James Watson and Francis Crick. Now, how does the replication of the DNA takes place? It takes place in a following steps, right? Number one is activation of nucleotides, right? Okay, now what is activation of nucleotide? Activation of the nucleotide is generally done by using ATP, right? ATP, that is, it is generally used by phosphorylase enzyme, right? Okay, so when you talk about activation of nucleotide, there are various nucleotides, right? 4 different types of nucleotide, that is deoxyribonucleoadenosine monophosphate, deoxyribonucleoguanosine monophosphate, deoxyribonucleocytosine monophosphate, and deoxyribonucleothymine monophosphate. Now they undergo phosphorylation reaction, that is, by using ATP in presence of phosphorylase, they generally undergo phosphorylation reaction. So they generally form deoxyribonucleotide triphosphate, that is D-ATP, D-GTP, D-CTP and D-TTP. Okay, so that is generally called activation of the nucleotide, which is generally done by phosphorylation reaction by using ATP in presence of enzyme phosphorylase. Second step is initiation or origin. Now, what exactly is origin or initiation? Initiation or origin is the point where the DNA generally undergoes replication. You can see this is a type of a point where the DNA is generally undergoing replication. So the point where the DNA undergoes replication is generally called as origin, or it is also called as initiation point, right? Now, when you talk about this origin in prokaryotic cell, there is a single origin or a single replicon, whereas in eukaryotic cell there are many replicon or many origin, right? Now, in a specific area where the origin starts, that is generally termed as O, and where the termination takes place, that is termed as T, right? So O stands for origin point and T stands for termination point. Third step is incision, the point where the DNA molecule is cut into 2 different strands by using the enzyme, that is replicating enzyme, that is restriction endonuclease, right? So now the DNA is generally cut at a specific point. You can see the DNA is generally cut at a specific point, and this incision is generally made by a very important type of a cleaving enzyme. That cleaving enzyme is generally called as replication. You can call it as restriction endonuclease, right? So restriction endonuclease is a kind of an enzyme. It is a kind of a cleaving enzyme which is going to cleave the DNA at a specific point. That is called incision. Fourth point is generally called unwinding of a DNA molecule. Now, after the incision, right, the two strands will start separating from each other. So the unwinding of a DNA molecule is generally done with the help of an enzyme DNA helicase, right? DNA helicase is a type of a unwinding enzyme which plays an important role in unwinding of a DNA molecule. Now, two strands are separated from each other. This strand will come in this direction and this strand will be separated in this direction. So two strands of a DNA are separated from each other by an enzyme. That enzyme is called topoisomerase. Topoisomerase is the enzyme which will start splitting the two strands of a DNA molecule. And two strands are stabilized with each other by a stabilizing enzyme called as single-stranded binding protein, SSBP, right? Single-stranded binding protein is a type of a enzyme which will generally stabilize or make the two strands of a DNA molecule stable. That is called unwinding of a DNA molecule. So unwinding of a DNA molecule takes place with the help of a DNA helicase enzyme. Two strands of a DNA start separating from each other by the activity of topoisomerase, and two strands of a DNA are stabilized with each other by the activity of single-stranded binding protein, that is SSBP. Now, fifth step, replicating fork. You can see once the unwinding of a DNA molecule takes place, now the two strands start separating from each other and it looks like a Y-shaped structure. It looks like a Y-shaped structure, so it is generally called as replicating fork. Okay. Now, sixth step says that formation of a complementary strand, right? Formation of the complementary strand. So for the formation of a new complementary strand, each separated strand acts as a template strand. Am I right? For the formation of the new complementary strand, each separated strand acts like a template strand or the old strand. Now what happens is that new nucleotides are being selected from the nucleoplasm, right? New nucleotide sequences are generally selected from the nucleoplasm, and a small enzyme is added, that is RNA primer is a small enzyme which is usually added at the 3' end of the template strand, right? So this is the 3' end of the template strand, that is the old strand, and there we are going to add a small enzyme. The name of the enzyme is RNA primer. Now what will happen is that new nucleotides are generally selected from the nucleoplasm, correct? Now this nucleotides will start pairing with the nucleotides. This nucleotide will start pairing, right, with the complementary nucleotides which are generally present on the template strand. This is the separated template strand, right? Now new nucleotide sequences will be selected from the nucleoplasm, and the nucleotides will start pairing with the complementary nucleotides which are generally present on the template strand. Thus there is a formation of a hydrogen bond between them, right? Now further, this nucleotides generally plays an important role in the formation of the phosphodiester bond, right? Now whatever the nucleotides which are been bounded with each other, there will be the formation of the phosphodiester bond. Thus what you can say is that there is a formation of a new strand, that is a complementary strand, with the help of an enzyme called DNA polymerase. DNA polymerase is a synthesizing enzyme which will generally play an important role in the formation of a new structure or a strand of a DNA. This step is generally called synthesis of a complementary strand. So I repeat once again, synthesis of a complementary strand. For the synthesis of a new complementary strand, every separated strand acts as a template strand or a old strand. A small enzyme called RNA primer is generally added at the 3' end of the template strand. Here, new nucleotides are selected from the nucleoplasm. Now, the nucleotides will start pairing with the complementary nucleotides which are present on the template strand. Correct? There is a formation of the hydrogen bond, and further each nucleotides will be bounded with each other by phosphodiester bond, and thus it leads to the formation of a new strand by a synthesizing enzyme involved called DNA polymerase. Seventh step is generally called formation of leading and lagging strand, right? You can see this is the leading strand and this is the lagging strand. The template strand with the free 3' end is called as leading strand or the continuous strand, and the template strand with the 5' end is generally called as the lagging strand or the discontinuous strand, right? So whatever the template strand which is generally going to have a free 3' end is called as leading strand or the continuous strand, and the other template strand having a free 5' end is called as lagging strand, right? Lagging strand or the discontinuous strand. Now, why this is called as discontinuous strand? Because you can see that the discontinuity of the strand is generally broken down due to the presence of a smaller fragment. Those fragments are generally called as Okazaki fragment. Correct? Okazaki was one of the first scientists who for the very first time observed that there are certain fragments on a lagging strand, and that's why he called them as Okazaki fragment. Later, this Okazaki fragments are joined with each other by joining enzyme called as DNA ligase. DNA ligase is a joining enzyme which is further going to join the entire strand of the lagging strand. That is, Okazaki fragments are joined by the DNA ligase enzyme, right? And finally, there is an enzyme called as DNA gyrase. DNA gyrase is a type of an enzyme which is now going to form a daughter DNA helically, right? So further, the entire helical DNA, daughter DNA will be generally formed with the help of a DNA gyrase enzyme, right? Okay, and the last step, that is formation, 8th step, formation of a daughter DNA. Formation of a daughter DNA. So I can conclude it that there is a formation of a daughter DNA. There is a formation of a daughter DNA or a new DNA molecule from the separated old strand or the template strand. As there is a formation of a new daughter DNA molecule from the old separated strand of a DNA molecule, this method of replication is called semi-conservative replication of a DNA. So semi-conservative replication of DNA is the method where there is a formation of a new daughter DNA molecule from the old separated strand of a DNA molecule, right? So these are all the different types of the steps involving in the replication of DNA. First, activation of a nucleotide. Second, initiation or origin. Third is incision. Fourth is unwinding. Fifth is replicating forth. Sixth is formation of a new complementary strand. Seventh is leading and lagging strand. And eighth is formation of a new daughter DNA. This is called replication of a DNA molecule, basically taking place in the eukaryotic cell. Thank you.